how exactly do dead, inanimate chemicals figure out how to wake up, organize themselves, and start making decisions? For decades, scientists pointed to the primordial soup, a warm, ancient ocean filled with the basic building blocks of life. But there's a fatal flaw with that model. A sprawling, global ocean is incredibly diluted. The odds of the right molecules randomly bumping into each other long enough to spark complex, self-replicating chemistry are virtually zero. This leaves us with a paradox. To build early genetic material, you need extreme concentration. You need a physical boundary, like a cell membrane, to trap those ingredients together. But you can't build a complex cell membrane until you already have the genetic instructions to do so. Without a way to concentrate materials before membranes existed, the math fails. Life shouldn't have started. The solution is a quirk of physical chemistry called liquid-liquid phase separation, or LLPS. Look at this oil and water mixture. The oil separates into distinct droplets, creating sealed compartments without physical walls. Atomically, hydrogen, electrostatic, and hydrophobic bonds pull dispersed molecules together, locking them into dense liquid crystals called coacervates. Floating in the primordial ocean, these coacervate droplets acted like natural vacuum cleaners. They swept up free-floating RNA, peptides, and proteins from the diluted water. By pulling them into highly concentrated microenvironments, the droplets radically accelerated reaction times. What was once a static, empty ocean became millions of active chemical laboratories. Fluid physics provided the critical first architecture for life to self-assemble. These liquid boundaries do more than provide structure. They create a mathematical tipping point. This sigmoidal curve plots molecule concentration against physical state. Below a threshold, molecules remain scattered, the off state. But cross that threshold and the system snaps. Molecules instantly collapse into a dense droplet, turning on. A chemical mixture is reading an environmental input, passing a threshold, and executing a distinct output. This is a binary switch, a primitive computational circuit. Molecules possess intrinsic molecular agency. Simply by following the laws of thermodynamics, they can collectively sense their environment and change their physical state in response. This physical transition provided the template for all biological decision-making. You can see this hardware running in single-celled organisms today. Take phototaxis, the ability of microbes like ancient halobacteria to navigate towards a light source. These bacteria lack eyes or a nervous system. Instead, when light hits them, they use phase separation to rapidly cluster specific proteins, like rhodopsin and chlorophyll, into dense sensory droplets inside the cell. By packing these molecules so tightly together, the droplet acts like an antenna. It amplifies the energy transfer from a weak photon of light, triggering an ion flux that steers the cell's physical movement. Through internal fluid physics, a blind, single-celled organism can collectively navigate its environment. When evolution finally built permanent cell membranes, phase separation moved inside the cell to become the hidden operating system. Many of the most intricate signaling networks in the human body are regulated by these temporary droplets. The Wnt signaling pathway, which tells our genes when to activate, is a prime example. This diagram maps the inactive pathway. The blue degradosome is a droplet built to trap and destroy proteins, keeping genes off. When a signal arrives, it dissolves. A new droplet, the signalosome, forms, migrates into the nucleus, and activates target genes. Modern proteins evolved specifically to manipulate this process. This model of galectin-3 shows a hydrophilic head and a flexible hydrophobic tail, allowing it to act exactly like those ancient oil and water boundaries to drive droplet formation. Human cells still rely on ancient droplet physics to process information and execute genetic commands. The spatial location of these droplets dictates the physical architecture of our cells. As this diagram shows, when condensates form in the cytosol, a cell maintains a rounded morphology. When they form in the nucleus and extracellular space, the cell adopts an elongated shape. This shape-shifting allows embryos to develop tissues and mediates how cancer cells adapt. As seen here, these internal on-off nodes wire into overlapping circuits. Positive and negative feedback loops allow the body to process information across millions of cellular compartments. Ultimately, life is a beautifully unified network, coordinating its complex behavior through the fluid boundaries of phase separation.